All right, we're diving deep today into validation versus verification, and especially how it all scales as a product moves through its life cycle.
Right.
Now, given that you're working with botanical and fungal products,
Yeah.
we know you need to factor in natural variability.
Absolutely.
And that definitely adds another layer of complexity to this whole thing.
It does.
We've got a stack of interesting sources here today.
Okay.
FDA guidance documents, scientific papers, even some insights from a conference on psychedelic drug development.
Oh, wow. That sounds...
And you might be thinking psychedelics.
Yeah. What's the connection?
What does that have to do with my work?
Right.
But trust us, there are some really cool parallels
Interesting.
when it comes to regulatory challenges and navigating the unknown.
Yeah, I can see that. When you're dealing with new and complex substances,
Right.
there's a lot we can learn from each other about how to ensure quality and consistency.
And ultimately, it's all about patient safety.
Yeah,
Right.
absolutely. That's the bottom line.
But how we get there,
Right.
that's where understanding validation and verification comes in.
Exactly.
So let's break it down.
Okay.
What's the core difference between these two ideas?
Sure.
Especially as they apply to analytical methods.
So think of verification as confirming we're doing things right. Like, are we using the correct tests? Is the equipment calibrated? Are we sticking to the procedures? It's about laying a foundation of accuracy from the very start.
So it's like double checking the blueprint before we start building.
Exactly.
Making sure every measurement is precise.
Yes, precisely.
Okay, so verification is about making sure we're following the plan. Validation, on the other hand, is about proving that the entire method or process is reliable.
Right.
And gives us the expected results consistently.
It's less about individual components and more about the big picture.
So verification is the micro view, validation is the macro.
That's a good way to put it.
But you mentioned that validation isn't a set it and forget it thing.
No, not at all.
How does validation change as a product moves through its life cycle?
Well, that's where things get really interesting.
Okay.
The level of validation needed changes depending on where we are in the product's life cycle.
Gotcha.
Phase 1, 2, 3,
Right.
and then commercial production.
So let's start with phase 1.
Okay.
What are the key validation expectations at this early stage?
In phase 1, it's all about establishing the basics. We're asking, is this method accurate? Is it precise? Can it consistently measure what we intend it to measure? It's about building that initial groundwork.
So we're not going full-blown validation just yet. More like establishing proof of concept.
Precisely.
Okay.
We're laying the foundation.
And then as we move into phase 2, we start fine-tuning and validating for specific parameters that are directly relevant to our product.
Yes.
So it's getting more tailored, more product-specific.
Okay. We're building on that initial validation from phase 1, gathering more data, refining the method, and ensuring it's robust enough to handle the specifics of our product.
And then things get real serious in phase 3, right?
That's right.
What's the validation focus at this stage?
Phase 3 is all about demonstrating that our entire manufacturing process, from raw materials to final product, is robust, reproducible, and scalable.
Gotcha.
We're proving we can consistently produce a high-quality product that meets all the required specifications.
It's like taking our finely tuned recipe and making sure it works perfectly.
Exactly.
In a large-scale bakery, not just in our test kitchen.
A perfect analogy.
We're demonstrating commercial viability
Right.
while maintaining quality and consistency.
That's the goal.
But even after launch, the validation journey continues, right?
It does.
What are the ongoing expectations during the commercial phase?
You're absolutely right.
Okay.
Validation is an ongoing process. Even after a product is on the market, we need to monitor the process, make adjustments as needed, and even revalidate if we make any significant changes to the formulation, manufacturing process, or even our analytical methods.
It's about ensuring that our product remains consistent, safe, and effective throughout its entire life cycle.
Precisely.
Now, let's add another layer of complexity here.
Okay.
Botanical and fungal products.
Right.
How does natural variability impact validation?
Well, that's where the real challenges lie.
Okay.
And frankly, the excitement. With botanicals and fungi, we're dealing with Mother Nature's inherent variability.
Right.
Unlike synthetic compounds, natural materials can fluctuate in their composition due to a multitude of factors.
Like what?
Like growing conditions, harvesting techniques, and the time of year.
So it's like trying to standardize a recipe
Exactly.
when your ingredients have a mind of their own.
That's a great way to put it.
This inherent variability directly impacts validation.
Yeah.
It does.
Our analytical methods must be robust enough to handle these natural fluctuations and still produce reliable results.
We need to be confident in our data.
So does this mean we need broader acceptance criteria for our methods?
It often does.
Okay.
We might need to widen the acceptable range for certain parameters to account for the natural variation we're bound to see in our botanical or fungal starting materials.
So instead trying to eliminate the variability, we're building our methods to accommodate it.
Precisely.
We're embracing the natural variation, understanding its impact, and designing our analytical methods to be adaptable and reliable within those natural fluctuations.
That's the key.
Now I'm starting to see why those insights from the Psychedelic Drug Development Conference are relevant here.
Oh, absolutely.
They're dealing with similar variability issues.
They are.
But in the context of therapeutic compounds, right?
Exactly.
So even though the end goal is different...
Therapeutic compounds versus consumer products. The challenges of standardization and validation are strikingly similar.
Yeah, the underlying principles are the same.
And the solutions often involve similar approaches.
Like what?
Meticulous sourcing of raw materials, sophisticated analytical techniques, and a deep understanding of the inherent variability we're working with.
It all comes down to that.
This highlights how crucial it is to have strong analytical methods in place from the get-go.
Couldn't agree more.
They're our eyes into the product, helping us understand its composition, identify potential issues, and ultimately ensure quality and safety.
Exactly. They're essential.
And it seems like this focus on robust analytical methods is even more crucial.
It is.
When we consider mutagenic impurities.
Absolutely.
Especially in the context of botanical and fungal products.
You're absolutely right.
That's a critical area where validation plays a key role in ensuring patient safety.
It's essential.
And it's a topic that deserves its own deep dive.
It certainly does.
So let's shift our focus now to the fascinating world of mutagenic impurities.
Okay.
What they are, why they matter, and how we can control them, especially when working with naturally variable ingredients.
That's a big one.
It is.
It's a whole other level of complexity.
It is, but it's a crucial one to understand.
Absolutely.
So let's dive in.
Let's do it. Let's dive into those mutagenic impurities.
Okay.
We're talking about substances that can actually cause changes or mutations in DNA.
And DNA is essentially the blueprint of life, right? So any alterations to that blueprint could have serious consequences.
They can. Some DNA mutations can lead to cancer.
Oh, wow.
Which is why controlling mutagenic impurities in pharmaceuticals, especially those derived from natural sources, is absolutely critical.
Okay, I'm starting to see the gravity of this situation.
Yeah, it's serious business.
But how do these mutagenic impurities end up in our products in the first place?
Well,
is it all about natural variability?
Natural variability can definitely play a role, especially with botanical and fungal products. Remember those fluctuations in composition we talked about earlier?
Yeah.
Well, those variations can sometimes lead to the presence of mutagenic compounds.
So it's like Mother Nature sometimes throws a wild card into the mix.
That's a good way to put it.
But mutagenic impurities can also sneak in through other means.
They can.
Sometimes they're byproducts of the manufacturing process itself.
Right.
Maybe a chemical reaction goes slightly awry, or there's an unexpected interaction between ingredients.
Exactly.
So it's not always about the source material.
Right.
It could also be about how we handle those materials during production.
Absolutely.
And that's why it's so important to have a comprehensive understanding of both our starting materials and our manufacturing processes.
You need to know what you're working with.
And to have the right analytical tools to detect these impurities.
Yes.
Even in trace amounts.
You got it.
And that's where those sophisticated analytical techniques we discussed earlier come in.
Exactly.
Things like HPLC and GCMS.
Those are our workhorses.
They're essential for separating the impurities from the main components of our product and getting a clear picture of what's lurking within.
Like having a powerful microscope that allows us to see those tiny, potentially dangerous particles.
It is. And just like with a microscope, the key is knowing how to use these tools effectively
Absolutely.
and interpret the data they provide.
That's where the expertise comes in.
Now, we've mentioned the ICH-M7 guidelines a couple of times.
Yes, those are important.
Can you remind us what those are and how they relate to controlling mutagenic impurities?
Sure. The ICH-M7 guidelines are essentially a roadmap for assessing and controlling mutagenic impurities throughout a product's life cycle.
Okay.
They provide a framework for identifying potential mutagenic compounds, evaluating their risk, and implementing appropriate control strategies.
So it's not just about saying no mutagenic impurities allowed.
No, it's more nuanced than that.
It's a more nuanced approach, right?
Exactly.
The guidelines recognize that not all mutagenic impurities are created equal.
Right.
Some pose a greater risk than others.
Absolutely.
And the level of risk can vary depending on a number of factors.
There are a lot of variables to consider.
So it's a risk-based approach, taking into account the specific properties of the impurity and how it's likely to be encountered in the final product.
Exactly.
And that's where the concept of phase-appropriate control comes in.
Yes, that's key.
Just like with validation, the level of scrutiny and control we apply to mutagenic impurities changes as our product moves through its life cycle.
We adjust our approach as we go.
Okay, let's break that down a bit.
Sure.
What does phase-appropriate control look like in practice?
In the early phases of development,
Okay.
the focus is on identifying potential mutagenic impurities and gathering data to assess their risk.
Gotcha.
We're essentially trying to get a handle on what we might be dealing with.
So it's about gathering intelligence, building our knowledge base.
Exactly.
And then as we progress through the development process, we refine our control strategies, collect more data, and demonstrate that we can consistently limit those impurities to acceptable levels.
It's an iterative process.
So it's a gradual process, becoming more stringent and targeted as we learn more about our product and its potential impurities.
We're constantly refining our understanding.
This sounds like a carefully orchestrated dance between risk assessment, control strategies, and data collection.
It is. It's a balancing act.
And this dance becomes even more intricate when you factor in the natural variability of botanical and fungal products.
Absolutely.
Because that variability can make it harder to predict exactly what impurities might be present and at what levels.
Right. You got it.
So our control strategies need to be robust enough to account for those fluctuations.
You need to be prepared for the unexpected.
So what are some of those control strategies? What tools do we have in our arsenal to tackle these mutagenic impurities?
Well, as we've already discussed, analytical techniques like HPLC and GCMS are essential for identification and quantification.
Okay.
They allow us to see what's there and at what levels. But we can go a step further and use in silico tools to help predict the mutagenic potential of identified impurities.
Wait, in silico tools, what are those?
In silico tools are basically computer models that can assess risk based on a substance's chemical structure.
Interesting.
They can help us prioritize which impurities to focus on based on their likelihood of causing problems.
So it's like having a crystal ball.
Kind of.
That can give us a glimpse into the potential risks of different impurities.
In a way, yes.
Okay.
They're incredibly useful for early screening and risk assessment.
This is fascinating. We're using cutting-edge technology to predict potential problems.
It's pretty amazing.
Before they even arise.
That's the goal.
And then, of course, we have experimental assays like the Ames test, which directly assess the mutagenic potential of a substance.
That's the gold standard.
So it's a combination of predictive modeling, analytical confirmation, and experimental validation.
We use all the tools at our disposal.
It's remarkable how much science and technology is involved.
It is.
In something that seems as simple as taking a herbal supplement. Or using a plant-based skincare product.
There's a lot going on behind the scenes.
It's true. There's a lot happening behind the scenes to ensure that those products are safe and effective.
Absolutely.
And it's all thanks to the dedication and ingenuity of scientists and researchers.
I agree.
Who are constantly pushing the boundaries of knowledge and innovation.
They're the unsung heroes.
Now, you mentioned earlier that not all mutagenic impurities are created equal. Some pose a greater risk than others.
Yes, correct.
What are some of the factors that influence the level of risk?
Well, several factors come into play, including the potency of the impurity. The dose a person is likely to be exposed to. The route of administration. The duration of exposure. And even individual susceptibility.
Okay, let's unpack those one by one.
Sure.
What do you mean by potency?
Potency refers to how strong the mutagenic effect is. Some impurities are highly potent,
Mm-hmm.
meaning they can cause mutations even at very low concentrations.
Yeah.
Others are less potent
Okay.
and require higher concentrations to have an effect.
So it's like a sliding scale of danger,
Right.
with some impurities being much more worrisome than others.
Exactly.
And just like with any potential hazard, we need to assess the level of risk
Absolutely.
and take appropriate precautions.
That's the key.
And dose plays a role too, right?
It does.
I mean, it's one thing to be exposed to a small amount of something,
Right.
but quite another to be exposed to a large amount.
The dose makes the poison, as they say.
Exactly.
The dose
right.
refers to the amount of the substance a person is exposed to.
Okay.
And obviously, the higher the dose, the greater the risk.
Like the old adage, the
That-
dose makes the poison.
Exactly.
Even water can be harmful if you drink too much of it.
That's true.
So when we're assessing the risk of mutagenic impurities,
Yes.
we need to consider the dose a person is likely to receive from the product
Absolutely.
and make sure it's well below the level that could cause harm.
That's the goal.
Okay, that makes sense.
Good.
So we've got potency and dose.
Mm-hmm.
What about route of administration? How does that factor into the equation?
The route of administration refers to how the product is taken or used.
Okay.
For example, a substance that's inhaled might pose a greater risk
Okay.
than one that's applied topically to the skin.
Because inhalation provides a more direct pathway into the bloodstream, right?
Exactly.
So we need to consider how the product is going to be used
right.
and adjust our control strategies accordingly.
We need to tailor our approach.
For example, if a product is intended for inhalation,
Mm-hmm.
we might need to implement stricter limits on certain mutagenic impurities.
That's right.
It's like having different security protocols for different entrances to a building.
I like that analogy.
Some doors might need more robust locks or surveillance systems than others.
Exactly.
Now, what about duration of exposure? How does that come into play?
Duration of exposure refers to how long a person is exposed to the substance.
Okay.
A short-term exposure might pose a lower risk
Okay.
than a long-term one.
Right.
Think about it. A brief exposure to loud noise is less likely to cause hearing damage than prolonged exposure.
So it's about cumulative effects over time.
Exactly.
The longer you're exposed to something, the greater the potential for harm.
That's the idea.
And the same goes for mutagenic impurities.
It does.
We need to consider how long a person is likely to use the product
Right.
and adjust our control strategies to ensure safety over that period.
We're looking at the long-term picture.
Okay, that makes sense.
Good.
But what about something like individual susceptibility? Isn't everyone's body a bit different?
You're absolutely right.
Okay.
Individual susceptibility refers to the fact that people can respond differently to the same substance.
Yeah.
Some people might be more sensitive to a particular impurity than others.
Okay.
This can be due to genetic factors, overall health, or other lifestyle factors.
So it's like some people are allergic to peanuts,
Exactly.
while others can eat them with no problems.
That's a good analogy.
And this individual variability
Right.
can make it challenging to establish safe limits for mutagenic impurities that apply to everyone.
It's a tough one.
So how do we account for that?
Well, it's definitely a complex issue.
Yeah.
One approach is to use conservative safety factors when setting limits, essentially erring on the side of caution.
Makes sense.
Another approach is to conduct studies in diverse populations
Okay.
to get a better understanding of how different groups might respond to the impurity.
So we're gathering more data to make more informed decisions.
Exactly.
It sounds like a constant balancing act,
It is.
weighing potential risks against potential benefits.
That's the nature of the game.
But it's a crucial balancing act,
It is.
one that ultimately ensures the safety and well-being
Right.
of the people who use our products.
Absolutely.
So we've talked about the various factors
Mm-hmm.
that influence the risk associated with mutagenic impurities.
We've covered a lot of ground.
Now let's shift gears
Okay.
and delve into the specific strategies we
Right.
can use to control these impurities.
These are practical.
Exactly. So what are some of the key tactics for keeping these mutagenic troublemakers in check?
One important strategy is careful selection of starting materials.
Okay.
If we can start with ingredients that are low in mutagenic impurities, we're already ahead of the game.
So it's about choosing our ingredients wisely,
Exactly.
like a chef sourcing the freshest produce for their dishes.
That's a good way to think about it.
And this is particularly important for botanical and fungal products,
It is.
where the variability of the source material can significantly impact impurity levels.
That's right.
Right, because Mother Nature doesn't always follow our quality control guidelines.
She has her own agenda.
So we might need to implement rigorous testing protocols.
We do.
For our raw materials to ensure they meet our standards.
Absolutely.
So it's about setting those standards high.
Yes.
And then making sure our suppliers can meet them.
That's key.
And we can also work with suppliers to implement good agricultural practices.
We can.
That minimize the risk of contamination.
That's a proactive approach.
So it's about collaborating throughout the supply chain to ensure quality from the very beginning.
Exactly. It's a team effort.
Now, what about the manufacturing process itself? Can we do anything during production to control mutagenic impurities?
Absolutely.
Okay.
Optimizing the manufacturing process can play a huge role
Okay.
in minimizing impurity formation.
Gotcha.
This might involve adjusting reaction conditions.
Like what?
Like temperature or pH.
Okay.
To favor the desired product over unwanted byproducts.
So it's like tweaking the recipe to make sure the cake comes out perfectly every time.
That's a great analogy.
We can also look at things like the order of addition of reagents, or the type of solvent used to minimize the formation of those pesky mutagenic compounds.
We're trying to control every variable.
It sounds like a lot of fine-tuning and experimentation to get it just right.
It can be.
And this is where process validation comes into play.
Exactly.
We need to demonstrate that our manufacturing process consistently produces a product that meets our quality standards.
Absolutely.
Including limits on mutagenic impurities.
That's a must.
So it's about proving that our process is reliable and reproducible. Even with those complex botanical and fungal ingredients.
That's the challenge.
And we might even need to incorporate purification steps into the process.
We might.
To remove any impurities that do form.
That's another layer of control.
So it's like adding a filtration system to our production line.
Exactly.
To catch any unwanted particles.
A perfect analogy.
And those purification steps can be tailored to the specific impurities we're targeting.
They can.
We might use techniques like chromatography, crystallization, or distillation to separate the good stuff from the bad stuff.
We have a lot of tools in our toolbox.
So it's a multi-layered approach.
It is.
Combining process optimization with targeted purification to ensure a high-quality final product.
That's the goal.
And this multifaceted approach is particularly important.
It is.
When we're dealing with the inherent variability of natural ingredients.
That's where it gets really interesting.
Right, because Mother Nature doesn't always make things easy for us.
She keeps us on our toes.
But with careful planning,
absolutely,
rigorous testing, and a bit of scientific ingenuity, we can harness the power of nature.
Yes.
While ensuring the safety and quality of our products.
That's the ultimate goal.
Now, let's talk about something that's been popping up in our research.
Okay.
The emerging field of psychedelic drug development.
Interesting.
It seems like there are some interesting parallels.
There are.
Between the challenges they're facing and the issues we've been discussing.
A lot of overlap.
It's like we're all navigating uncharted territory together.
It feels that way.
Learning from each other's experiences.
Absolutely.
And this cross-pollination of ideas and knowledge is incredibly valuable.
I agree.
For example, the researchers working with psychedelics are developing innovative extraction and purification techniques.
They are.
To isolate the active compounds from plant material.
It's cutting-edge work.
And those techniques could potentially be applied to other botanical and fungal products, right?
Like
the same principles of separation and purification apply.
They do.
Whether we're working with psilocybin from mushrooms, or a specific medicinal compound from a plant extract.
The fundamentals are the same.
It's amazing how these seemingly disparate fields are converging.
It is.
Driven by the shared challenges of working with complex natural substances.
It's a common thread.
And the regulatory landscape is evolving alongside these scientific advancements.
It has to.
The draft guidance document from the FDA on designing clinical trials for psychedelics is a prime example.
That was a big deal.
Ah, yes. That document caused quite a stir in the research community.
It did. It was a game changer.
And for good reason.
Right.
It's the FDA's first official acknowledgement of the potential therapeutic benefits of psychedelics.
A historic moment.
So it's a sign that things are shifting.
They are.
That these substances are finally being taken seriously as potential medicines.
It's about time.
And the guidance document provides a framework.
It does.
For conducting rigorous clinical trials. To evaluate the safety and efficacy of these substances.
That's essential.
Which is crucial for gaining wider acceptance.
Absolutely.
And ultimately making these therapies available to the people who need them.
That's the goal.
Now, one of the things that struck me about the draft guidance document was its focus on patient safety.
Right.
particularly in the context of the psychedelic experience itself.
That's a key consideration.
Because the psychedelic experience can be intense and unpredictable, right?
It can be.
And that's why it's crucial to have trained professionals involved throughout the process.
Absolutely.
Guiding the patient and ensuring their safety.
It's essential for a safe and positive experience.
It's like having an experienced mountain guide.
I like that analogy.
When you're climbing a challenging peak,
right,
you need someone who knows the terrain, understands the potential risks,
yes,
and can help you navigate the journey safely.
That's a great way to put it.
And just like with mountain climbing, the preparation and support system are essential.
They are.
For a successful and positive outcome.
That's the key.
So we've got the scientific side covered, the rigorous analysis, the careful control of impurities,
yes,
the development of standardized extraction and purification techniques.
Right.
And now we're seeing the regulatory side catching up.
It is.
Providing guidelines for clinical trials.
Yeah.
And prioritizing patient safety.
It's encouraging to see.
It's an exciting time.
It is.
To be witnessing this convergence of science, medicine, and regulatory thinking.
That's a paradigm shift.
And it all comes back to the fundamental goal of harnessing the power of nature.
Absolutely.
To improve human health and well-being.
That's what it's all about.
Exactly. Whether it's through botanical medicines, fungal extracts, or even psychedelic therapies.
Yes.
We're exploring the vast potential of the natural world to heal and transform.
It's an incredible journey.
And this deep dive has given us a glimpse into the intricate web of science, technology, and regulation.
A complex interplay
that underpins this exploration.
It's fascinating.
It's a reminder that even the seemingly simple act of taking a natural product is a testament to human ingenuity.
It is.
And our ongoing quest to understand and utilize the incredible complexity of the natural world.
We're just scratching the surface.
It's fascinating how this field of psychedelics challenges those traditional ideas we have about drug development and regulation.
It really is.
It's like they're forcing us to think outside the box and consider completely new approaches.
They don't really fit the mold of your typical pharmaceuticals.
Wow.
The psychedelic experience itself, that journey, that's a core part of how they work as therapies.
Right.
That adds another layer of complexity we don't usually deal with.
It's not just about taking a pill and waiting for symptoms to go away.
Right.
It's about a guided experience.
Exactly.
Exploration of consciousness.
Right. And that kind of therapeutic approach requires a different mindset, both for the clinicians working with patients and for the regulators overseeing it all.
And we're seeing this shift reflected in that draft guidance document from the FDA.
Absolutely.
There's a real emphasis on the therapeutic relationship.
Yes.
The setting and how the patient integrates their experience afterward.
It's a more holistic approach than we often see, acknowledging that mind, body, and environment all play a role.
You were right when you said this field is pushing boundaries.
It is in so many ways.
It's not just the science of the compounds themselves.
Right.
It's about the human experience of healing and transformation as well.
And that's what makes it so exciting.
Yeah.
We're potentially at the forefront of a major shift in how we approach mental health and well-being.
It feels like we're rewriting the rule book, challenging those assumptions we've held for so long about what treatment looks like, and exploring new possibilities.
I agree. It's a whole new paradigm.
And those explorations come with challenges, of course.
Of course, there are always challenges when you're breaking new ground.
But the potential benefits are enormous.
They are.
Think about the research emerging on psychedelics, offering hope for those struggling with conditions like depression, anxiety, PTSD, even addiction.
It's groundbreaking research.
Traditional treatments haven't worked for them, but this could be a breakthrough.
It could be a game changer for so many people.
It's like we're opening up a whole new toolkit for addressing mental health.
And as we learn more, who knows what we might discover about the human mind itself.
We've covered a lot of ground in this deep dive.
We have. It's been a fascinating journey.
From validation versus verification
to
the complexities of botanical and fungal products, and then into this fascinating world of psychedelic drug development.
It's all connected.
It's been a journey.
It has a reminder that for all the science and technology involved, the core of it all is about ensuring those products are safe and effective.
Right.
That the potential benefits outweigh the risks.
And it's a reminder that nature, in all its complexity and variability, holds incredible potential for improving human health.
It's a powerful force.
We just need to approach it with respect, a healthy dose of caution, and a lot of scientific rigor.
That's well said. It's about working with nature.
Right.
Not against it.
Yeah.
Understanding its nuances, adapting our approaches, and using our knowledge to ensure quality and safety every step of the way.
So for you listening, here's one final thought to consider.
Okay.
We've talked about the challenges of standardizing these naturally variable products.
Right.
But what if that variability itself holds the key to even greater therapeutic potential?
Interesting question.
What if embracing that diversity rather than trying to eliminate it
allows
us to create even more personalized and effective treatments? It's a question worth pondering as we continue to explore the intersection of science, nature, and human health.
A lot to think about.
It is. Thanks for joining us on this deep dive.
It's been a pleasure.
We'll be back soon with another fascinating topic.
Looking forward to it.
Until then, keep exploring, keep questioning, and keep pushing those boundaries of knowledge.